So this is my DNA sequencing project for biotechnologies. And the first thing I'm going to talk about is the history of DNA sequencing and how it was developed. And the first major contribution was in 1953 by Watson and Crick, which is when they discovered the three-dimensional structure of DNA. And this discovery later in 1977 led to researcher Frederick Sanger to develop a more efficient technique for DNA sequencing, which surpassed any previous research and actually led him to get a Nobel Prize. This technique of Sanger sequencing involves determining order of bases in each strand of DNA, and by using purified DNA polymerase enzymes to synthesize DNA chains, you can make them into various lengths by elongating them so that you can create a specific DNA sequence that you would like. When DNA chain is elongated to the desired length, a terminating base, which consists of deoxynucleotide triphosphates, are able to end the chain because they lack the 3' hydroxyl group needed to form the phosphodiester bond between one nucleotide and the next. And after the DNA fragments of the different lengths are created, each individual fragment is then moved through a gel matrix as an electric field is applied. And as it moves through the matrix, the fragments are then separated in order of shortest to longest length. And this technique is called electrophoresis. So, like I said, when the DNA strands emerge from the capillary gel electrophoresis process, it goes in order from smallest to largest fragments, and it builds up one nucleotide at a time so that the DNA sequence can be formed and then later on read through peaks in a chromatogram. And next, I'm going to talk about how DNA sequencing is used in the past, present, and in the future. Firstly, in the past, it was discovered as a way to investigate different diseases, genetic illnesses, and mutations, which are caused by faulty genetic sequencing. And this allowed scientists to help facilitate treatments when there is a DNA mutation so that they could warn the individual about the mutation and possibly be able to create a treatment plan so that the mutation wouldn't affect them in their lifetime. And it was also discovered as a way to help molecular biologists understand gene sequences so that they could understand how the proteins function in certain systems and pathways of the body during natural DNA processes so they could apply it to the genetic sequencing process. Also, in the present, we are using an orthogonal method for confirming sequence variants identified by next-generation sequencing, which is going to replace Sanger sequencing in the future. So scientists are using this NGS testing to perform and perfect their methodology, since some regions that are rich in DNA in the body are resistant to sequencing because of amplification and capture in the ratio for DNA to the electrogel matrix is uneven, so the DNA normally takes over, so they need to perfect or redesign this technology. It is also used in the CRISPR technology, which genetically modifies embryos so that they could have very specific characteristics and attributes. And finally, it is presently being used so that in genome mapping, so that you can pinpoint specific mutations in sequences and be able to detect and possibly treat diseases before it is able to affect the individual. And finally, in the future, DNA sequencing, as I mentioned earlier, is going to be using next-generation sequencing, which will take over the Sanger sequencing because it has 100-fold improvement over Sanger sequencing, and it is able to, or will be able to in the future, detect a wider variety of genetic abnormalities such as single base pair substitutions, DNA rearrangements, and copy number alterations, which Sanger sequencing can not currently detect at the moment. And lastly, the next generation sequencing will end up costing more in startup fees and in laboratories and materials needed to administer these more advanced testings, but it will actually work out for the better because you'll be able to detect more as well as you can pay a bit more, so the ratio ends up equaling itself out. Next, I'm going to be talking about the societal and ethical implications of DNA sequencing, both the beneficial effects and the harmful effects. So first, for beneficial effects, DNA sequencing has allowed scientists to categorize the specific and important proteins in their specific pathways of the body that is used during natural DNA replication. And this has been a therapeutic discovery because it can allow computational biologists to discover even more treatments for these diseases and mutations, and give possibly in the future an opportunity to even be able and stop mutations before they can affect anyone. Next, DNA sequencing also gives an edge for forensic examiners because it helps them be able to link criminals to the crime by the gel electrophoresis, because it can show bands of DNA in criminals' fingerprints so that the fingerprint DNA can be matched to the criminal who committed the crime. And lastly, for the beneficial effects, DNA sequencing is able to detect parent strands of DNA and genes passed through generations, so it can link individuals with their ancestors and give a more well-rounded prediction of their ethnic backgrounds and who their lineage connects them to. So for the harmful effects, DNA sequencing is being used in the CRISPR technology, which, like I mentioned earlier, allows scientists to genetically modify embryos and give them very specific mental and physical attributes so that they are creating a perfect gene sequence for the child, which I think is very harmful because it creates children in an unnatural way and kind of makes them less immune to specific diseases, which isn't what is supposed to be happening naturally in the world. Another harmful effect is when genome sequencing is performed in utero on an unborn child, it can detect possible mutations that may develop in their lives, but it may cause a parent to unnecessarily worry, because just because a mutation presents itself in your DNA sequence doesn't necessarily mean it will develop during your lifetime. It is just a chance, but there are steps that you can take to make yourself less susceptible to contracting these diseases that your mutations allow you to possibly develop in your lifetime. And the last harmful effect is that, as I mentioned in the beneficial effects, DNA sequencing can link ancestry and ethnic backgrounds, but this can lead to the harmful effect that one ethnic group would believe that they are superior to another based upon number of mutations in their genes and what diseases they're susceptible to. Just because you may have more mutations than another ethnic group does not make you less than anyone else. So we wouldn't want this to lead to any hurtful or racial comments towards a specific ethnic group. So here is just a few pictures. In the top left corner would be how the CRISPR technology sequences the DNA for embryos and modifies it to their likings. And then on the right, we have how forensic examiners use the DNA bands to connect fingerprints to the criminal who committed the crime. And lastly, on the bottom, we have a few statistics of how DNA sequencing can give people warning of diseases that they may contract. This one is about cancer. And so people who had the pre-warning that they might have the mutation that would give them cancer can better prepare themselves and maybe make a few changes in their lifestyle so that they could be healthier and maybe be able to not develop this disease at all, or help them cure it faster because they already knew that it possibly could come. Now I'm going to talk about the natural DNA replication versus DNA sequencing process. So for the natural replication, the enzymes DNA helicase and topoisomerase work together to unwind DNA strands by cutting